Hello.
Hi Serena, how are you?
Good, good.
Hi Z.
So excited. So excited to hear about astrocytes.
Yeah, me too. I'm very excited. Hi Moritz. Am I saying your name right? Or Moritz? I'm not sure. Moritz. How are you?
Hello, Moritz. Your mic is still muted. In the lower right, there's a microphone you could unmute. Can you hear us?
If you're on your phone, it's all the way on the bottom right hand. There should be a little microphone symbol, and if you press on it, you should be able to unmute. Oh,
Yes.
there we go.
Yeah,
Hey.
I'm so sorry. I had problems with the output first.
Oh, no worries. Welcome.
Okay, we can hear you now.
How are you today?
Yeah.
Hi, Victoria. How are you today?
Say it again. Sorry.
Oh,
Hello, hello everyone. I'm well, thank you. Nice to see you, Katarina, Serena, Moritz.
Welcome, welcome. We have an astrocyte room. It's my favorite
Ah,
topic.
finally. So excited. I'm so happy. Thank you. This is why we've been doing this. The whole reason is for this astrocyte room.
our whole life led up to this moment.
No, Maurice, no kidding. Almost every chance I get, if the topic is even, if I can see an astrocyte angle, I go there.
That's exciting to hear. Astrocytes get a weird fall. Not weird. Get sort of an eclectic mix of people interested in it.
Well, I've really gotten the sense that there's been such a deep, renaissance of their function in the last, what, decade or so. Would you say that?
Yes, to some extent. And I think to some extent, sort of, there's still a lot missing. Sort of, there's, for example, I mean, there's a lot of work has been done sort of on like gliotransmission, where astrocytes release various factors, release neurotransmitters, release glutamate, release a lot of other things, and sort of there's enough been done to show that they do it, but very little is advanced on sort of like understanding what triggers it, how it gets released, where it gets released. Sort of a lot of those tools have really sort of stagnated to some extent in the last decade, I would say.
Oh, really? Because I mean, I've taken such an interest in calcium waves throughout the syncytium and how they may contribute to so many higher level processes in the sense of, a sort of dialogue between neurons and these waves in the syncytium. I'll be really interested to hear your work and get a good discussion going.
Yeah, yeah, but so, I mean, for example, I think astrocyte calcium hasn't really advanced much. I mean, sort of, there's been really nice work on the calcium waves and the calcium sparklets and out in the processes, but sort of really connecting that to like what comes out, it's sort of all very crude. Of you have some sort of like secondary measure of some gliotransmitter, and you sort of vaguely correlate it to calcium, but sort of everything gets vaguely correlated to calcium, and there's no real like nobody's sort of figured out really exactly what triggers these calciums, or like what's downstream of it. Like, how do you go from a calcium sparklet or a calcium wave to D-serine getting released, or ADP getting released, or something else? I mean, it's sort of, I think it's been the limits of, we have great calcium tools, which makes calcium easy, but we don't really have or haven't had the tools to sort of go after the like what comes next.
Hmm. Well, and do they, and I also look at the whole tripartite synapse, and like curious and about, do these waves then, there's some reports of, potentiating postsynaptic neurons in long-term potentiation, or is that, I mean, are these waves somehow driving neural synchrony like in other reports, or what's really going on there?
Yeah, and I mean, there's definitely really good work on it to some extent, but sort of, I think it hasn't, sort of coming from like the neuronal side, sort of where there's been so much work, and sort of like we really know like what a synapse does and how it works, of calcium entry, vesicle release, or like everything sort of connected. And I mean, we don't understand everything, but sort of like we at least know the like pathway for everything. Mm. And I think sort of like there's just sort of like key steps missing in the pathways. So I mean, for example, I mean, one of the interesting things on astrocyte calcium is when you stimulate neurons, you don't get a repeatable astrocyte calcium signal, which is sort of like completely different from like any like neuronal function we look at. And sort of it makes sort of like calcium, astrocyte calcium signaling on a really like mechanistic level hard, because you just can't like give the same stimulus and get the same calcium signal out. It just becomes, there's sort of clearly something in between that we don't really understand
Yeah, they're kind of moody, aren't they?
yet. Exactly. I mean, sort of, there's clearly a lot of calcium activity going on, but sort of, sort of, to me, it always seems like it's hard to make tractable if, I mean, if I can't evoke it the same way repeatably, it's sort of like it becomes much more difficult to study.
Well, so doesn't that suggest that there's timing and context there that we're
Yeah,
missing?
yeah, exactly, exactly. I mean, I think a lot of it is probably context. Sort of, we don't really know what state the astrocytes are in. That clearly matters for if you're going to evoke a calcium wave or not, or a calcium spark or not. But till we have like a tool to sort of really like at least define what an astrocyte state is in that sort of sense, it, or at least nobody's really like, even as far as I know, concretely found a way to go after that.
Well, you're being very cautious, and I certainly respect that.
Well, I mean, it's biased by my own interest and my own sort of like ideas of what I think is tractable for my projects and my experiments is also the thing of like there's a lot of interesting and mm, really novel stuff on like astrocyte calcium with in vivo and with behavior, which is not exactly my expertise is for in terms of like experiments and projects I do, which is also sort of just biases me towards I like when I have controlled situations where I can stim and where I can get repeatable things that I can sort of manipulate and try to dissect.
Right, it can easily be a slippery slope, and sometimes in Clubhouse, the conversation wanders from me. We like to keep a high standard of science here, but then, we'll get off into discussions of behavior and then philosophy and the hard problem of consciousness, and it all goes everywhere. But I'm really excited about this topic, and I guess we can start to get started here pretty soon. Um, yeah.
Yeah, let's start. So thank you for the pre conversation. It was really already interesting. So thank you for that. And because sometimes, usually, this waiting time can get kind of boring, but you guys made it interesting. So perfect.
I couldn't resist.
So yeah, welcome everyone to Science Society, and a special warm welcome to Moritz here. And let me introduce you to the audience a little bit before we start. So Dr. Moritz Armbruster, he's a research assistant professor in neuroscience at Tufts, and he did his Bachelor of Science at Cooper Union and his PhD at Rockefeller University. And his research areas of interest are focused on astrocyte-neuron interactions and how these astrocytic functions can modulate synaptic and somatic neural function. And he has found novel neural control of astrocytic glutamate clearance kinetics with synapse specificity and fast kinetics. And in order to investigate or answer some questions he has around that, he uses a combination of astrocyte and neural electrophysiology and cutting edge live imaging of glutamate, calcium, voltage, and other biosensors. So yeah, Moritz is doing really amazing research. And before you start with basically with the talk about this, your ongoing research and the latest or the publication we mentioned, Victoria usually asks our guest speakers like a couple of more general questions, if that's okay, like a
Yes,
short interview, and so that the audience gets to know you as a researcher maybe a little bit more before we go like to the hard science, if that's okay with you.
sure.
All right, here we go. Thank you, Katarina, for that fantastic introduction. And Moritz, welcome. Science Society welcomes you. We are so happy that you're here to discuss astrocytes with you. You have no idea. Well, maybe now you have a little bit of an idea. Yeah, to help carry us into your discussion about your work with astrocytes, I would like to ask you if you can look back through your life and reflect on a time that you noticed that you felt an affinity towards science. And that could be maybe when you were a child, maybe a relative, or family tradition, or maybe some part of your education, but sometime that you noticed that you felt really connected to science.
I mean, I've been interested in science for a long time, even growing up. I mean, it varied a lot in the directions of science, and in growing up, I would never have predicted that I would have gone into biology. Sort of biology was the least interesting to me. I was much more on these sort of like engineering, mechanical type things. Yes. I mean, what really got me into biology and interest in biology was a professor in undergraduate, and that really excited me. And then I started sort of like going more to sort of lectures at Rockefeller, like on the side, and there are sort of a few presentations looking at sort of the noise and stochasticity in biological systems really got me excited and really like sort of pushed me towards switching over from more of an engineering background to towards biology and neurobiology specifically.
All right, thank you for going there with us. And it's interesting to hear how many people do switch their fields of interest, and so then beyond that, can you walk us through the path that has brought you to your current research? Maybe some areas of interest that will bring us to the research that you're doing today.
So my sort of like scientific and biological background is, I mean, I did my PhD with Tim Ryan at Rockefeller, and it was sort of highly focused on presynaptic neuronal function, but also highly focused on using optical techniques, using whatever fluorescent sensors we could get our hands on and make work to really dissect like fundamental mechanisms controlling the vesicle cycle. And sort of that really sort of set a lot of my approach to science, of like really like try to get at the mechanistic and try to sort of find the best tool for the job. And when I moved to Tufts, sort of I really sort of switched from the neuronal side to the astrocytic side, and it seemed like there was at the time new sensors and new techniques coming out that could really start to open up new areas of investigation. And Tufts has a strong core of researchers interested in astrocytes, which really helped and really drove my interest there. And that's sort of like combining those is sort of like how I come to sort of the type of research that we're going to be talking about today, of sort of trying to utilize new and cutting edge sort of fluorescence and imaging approaches to piece out new ideas and new biology in neurons and astrocytes.
Well, thank you very much. And at this point, you're welcome to begin your presentation, and know that if you would like to have your Q&A following your discussion, then that's great. And if you'd like to have the Q&A sort of driving your discussion along the way, then that's entirely up to you. And we will take care of bringing guests up to ask you questions and navigate questions that are in the room chat. And you can just relax and discuss.
Okay, sounds good. And I mean, absolutely feel free to break in and interrupt with questions. I think that is always helpful and useful in terms of driving the conversation.
Great, and in addition, I just would say to friends that do come up to interrupt and ask Dr. Armbruster any questions, to please flash your mics first so that we can call on you and make sure that everybody's voice is heard. So thank you very much, and the mic is yours.
Okay, thank you. And thank you for inviting me. And so today I wanted to talk about our new study on looking at depolarizations in fine astrocyte processes. And this is sort of something that we've sort of been building up for a while. But I think before I go into the meat of the astrocyte biology that we discovered, I wanted to give a quick introduction to sort of the techniques and the tools we're using, because I think these genetically encoded voltage sensors that have been developed in over the last 10 years are really exciting tools, and I think have really started to reveal new biology that wasn't accessible before. So in our study, we used two different voltage sensors. One is called ArcLight, which was developed in the Pirabone lab at Yale, which essentially utilizes a voltage sensing domain from a voltage sensitive phosphatase tied to pH sensitive GFP. While the other one is a completely different molecule. It's it's called Archon, and it's derived from an archaerhodopsin proton pump that Adam Cohen at Harvard figured out that in addition to the optogenetic use of it for pumping protons, it had a completely separate voltage sensitive fluorescence in the far red. And these have been further developed. The Archon version that we're using was further evolved by Ed Boyden at MIT. And what they really provide is they now provide a method to, in a highly localized and fast manner, we can start imaging the voltage of a neuron or of an astrocyte or of any other cell type. And what this really has enabled is we can start getting at subcellular voltage changes. Like one of the first uses was sort of comparing the action potential waveform at a soma versus far out on the axon in the synapse. And that there were clearly differences, and that this could drive different behavior at the synapse that would normally go undetected by traditional electrophysiology, because normally we patch a soma, and rarely and in very low throughput do people patch presynaptic boutons or postsynaptic structures. So this really sort of these two tools, and these are by no means are the only ones. In the last decade, there's like every year several new versions have been published, and they all have sort of their strengths and weaknesses. So Archon is very dim by fluorescence standards, while Arclight sort of is not as good kinetically as Archon. Archon can really follow an action potential. Arclight is a little slower. So they each have all still, they're not perfect is the short answer, and they're not quite as easy to use as gcamps these days, but they're constantly being developed, and I think these are really exciting tools. So what really drove our interest in starting to voltage image astrocytes was this finding that Katerina alluded to, of where we found that astrocytes dynamically seem to control glutamate clearance. So as a bit of background, sort of astrocytes have these really fine processes that a lot of the time wrap around synapses, and these are packed full of, one of the things they're packed full of is glutamate transporters, so that when excitatory synapses release glutamate, in the astrocytes have the transporters to take up the glutamate before it activates too many things. And sort of if you don't have it, you get essentially seizures from overactivation. And one of the curious things we found was then neuronal activity would control the rate of glutamate clearance, but that it was really the amount of stimuli and not the amount of glutamate. And this was sort of something really puzzling for us because none of it really made a lot of sense of how is the neuronal activity slowing a glutamate transporter on an astrocyte, and especially if it's not the amount of glutamate. So that had us sort of scratching our head for a long time, and we tried many different things, and pretty much the only hypothesis we could come up with was that these astrocytes must be locally depolarizing because these glutamate transporters are highly voltage dependent. Now, the thing that sort of made the hypothesis feasible is that astrocytes are rather funny cells in terms of electrophysiology. So we and others have patched astrocytes for a long time, but contrary to neurons, they are very leaky. And so what I mean by that is in a neuron, when you patch a neuron and you can voltage clamp it at whatever voltage you want, you can pretty much voltage clamp most of the neuron. That the membrane resistance is high enough that when you control the soma, you control a large space, sort of the space clamp of the cell. While astrocytes are extremely leaky. So there was a really nice paper that did the heroic experiment of dual patching a single astrocyte soma, and they found they couldn't voltage clamp one side of the soma to the other. And sort of, it turns out you have a space clamp of 3 microns about. So what that really means is that when we patch the astrocyte soma, the voltage record there can be way, way, way different than what's happening out in the processes. And these processes are the sites where they really interact with the neurons, and where we think is sort of the key element for a lot of the neuronal function, the neuronal astrocyte interplay and interfunction that they do. So that is what really drove and pushed us to sort of try to voltage image astrocytes and gave us hints that there might be something exciting there. So in our paper, we sort of first expressed these 2 voltage sensors in astrocytes using AAV, so adeno-associated viruses, sort of viral tools to express these sensors in a cell type specific manner in mice in the cerebral cortex. So everything we did essentially focused on the cortex, and specifically the somatosensory cortex, just because it's the system we have the most experience with and is well established in our lab. And sort of the first thing we discovered that sort of really jumped out and kept us really hooked that we had something exciting was when we looked at the kinetics of our voltage imaging signal versus the signal when we would patch clamp an astrocyte, our voltage imaging signal would decay about 10 times faster. So when you patch clamp an astrocyte, you get a depolarization when you activate neurons. So all of this, sorry, to sort of clarify, all of this is where examining an astrocyte and activating axons that come into the astrocyte's domain. So it's sort of triggering neuronal and synaptic activity in its domain to essentially evoke glutamate release, to evoke anything else that these pretty much excitatory axons are going to release and that the astrocyte is going to respond to. So in this sense, sort of like the first thing that really jumped out was that the decay kinetics were pretty much an order of magnitude faster. And this really, at least, was getting close to sort of what we had found in our previous like glutamate study, that sort of everything in our glutamate study sort of recovered on a really fast time scale, even faster than what we saw here with the voltage imaging. But that gets into really fine details, but sort of it at least sort of checked that we were on the right path. And we sort of started by looking at the kinetics because mainly because sort of the amplitude and sort of calibrating the amplitude is a challenge, and just sort of remains one of the big challenges with voltage imaging. But from there, sort of we moved on to trying to figure out exactly like when we get this fluorescent voltage signal from our astrocytes, where exactly is it coming from? Is it sort of dispersed? Is it coming from the soma? What's sort of the spatial component of it? And this is sort of part of the project that sort of took a long time and sort of like a lot of challenging analysis to convince ourselves that what we were seeing was really true, and that we would believe it ourselves, and that we could sort of trust it. And sort of we tried to sort of challenge it with many sort of different approaches. And sort of what we found is that the majority of the signal was coming from very small hotspots, so on the order of a micron or less, and really sort of pushing the limits of like the spatial resolution of the microscopes we're using. And we did sort of a lot of work of sort of trying to like overlay it to try to figure out the really precise, as precise of a spatial component as we could. And what we sort of found was that and we get sort of these hotspots that are oblong, with the short axis being on the order of half a micron and the long axis being on the order of 1.5 to sort of 2-3 microns. And sort of what we really think is going on there is that this really could be like a fine astrocyte process that is sort of touching a synapse. That is sort of, you really get the like end of the process, get a really focal depolarization there, and then due to the about 3 micron space claim, you get a bit of a propagation of the depolarization along the process. And that's why you get this sort of like oblong shape. So sort of to give a little background on sort of astrocyte morphology, is that astrocytes have, I sort of always term them fuzzballs, because they have a fairly small cell body, and then are extremely ramified with lots and lots of fine processes that go honestly beyond the diffraction and beyond what we can resolve. So by EM, the like fine astrocyte processes that like touch synapses are on the order of 100 nanometers or so, which is well below what we can resolve. But sort of, and that's sort of the like caveats I'll throw in of, we're really pushing the limits of what we can resolve in these spatial, and sort of these spatial hotspots, the size, are probably filtered by our microscope, our point spread function. So I don't want to really say that they're precisely half a micron by 3 microns. It's more that they are really small and pretty much as at the limits of our resolution where we can really put a size on it. But what is clear is they don't really happen at the soma. They really happen, they don't even really happen at the primary processes. So for this, we double labeled the astrocytes with a cell fill with the TT tomato, and this is so at the top of figure 3, where you can really see the soma, the primary process, and then all these sort of like hotspots, and they're pretty much excluded from the primary process. They're excluded from the soma, and they really, everything we can do really points to them being at or close to the synapse.
So, quick question.
Yes.
So, what from what I'm getting here is your at least the thinking behind this is that the synaptic activity, which is on the length scale of what 100 nanometers or so, is initiating these depolarizations, but they're spreading out microns and sort of like the from the leaves into the branches, as it were, but not to the trunk or anything necessarily.
Yeah, exactly. So uh, it, that part gets a bit more complicated, and I can touch on that later. But sort of, it's essentially that is the idea that the depolarization sort of initiate with a synapse, and then sort of you get a spread onto essentially down the process, and sort of it falls off on the order of a micron, few microns. That's exactly sort of how we're picturing it. And I mean, there's with the caveats of like exactly how fine these are, of like we can't really resolve the finest processes, honestly. But sort of, and sort of like one of the things that we really did to sort of like convince ourselves that we're that this is sort of the case is if we put in sort of a second stimulator and activate a different pathway, sort of we get completely non-overlapping depolarizations. That's sort of like if we activate one set of synapses, we get one set of depolarizations, and if we activate a second set of synapses, we get a different one. Sort of really suggesting that this is synaptic and really like highly localized synaptic. But then, sort of like as you sort of alluded to, it was sort of one of the big questions is sort of like exactly what on the synapse is driving it, and sort of the other really big question is sort of like it's nice to sort of see a depolarization, but the really significance comes when we can put a number on sort of how big is this depolarization, because so we know from our patch clamp studies and other people's patch clamp studies, if you record at the soma, you get a 1, 2, 3 millivolt depolarization or so, which all in all isn't much to get excited about. So the other electrophysiological property of astrocytes is they're not just really leaky of low membrane resistance, they're fairly permeable to potassium, which sets their resting membrane potential. So they're very hyperpolarized. They rest around minus 80 to minus 90 millivolts, so close to the potassium equilibrium. And sort of the one of the things we did was sort of we went through an extensive sort of calibration to sort of try to put a number on how big these like hotspot depolarizations are in terms of amplitude. And we did this by essentially washing on different higher potassium solutions to sort of depolarize everything to sort of known levels. So like we can patch clamp and see that adding 10 millimolar potassium to the bath is going to depolarize it by 20 millivolts. Adding 5 is going to polarize it by 10 millivolts. And sort of using that as a calibration, doing that by EFIS and by voltage imaging, we got a calibration that puts our hotspots at pretty close to a 20 millivolt depolarization, which is sort of compared to the soma that gets about a 2 millivolt depolarization is huge, and sort of I think way bigger than what anybody really pictured an astrocyte doing, because everybody has sort of thought that astrocytes are largely passive electrophysiologically. There's a few currents like glutamate currents, but it doesn't really change their membrane potential much. That's at least been the thought. And sort of seeing that we were getting hotspots of 20 millivolts really convinced us that sort of our initial thought that this could actually be modulating glutamate clearance is potentially feasible, because now these transporters see a 20 millivolt change, but it also sort of really highlighted the excitement that this voltage imaging approach could really show us something new that we hadn't seen before. So then we get to the sort of question of what really is driving these changes, and sort of we start to try to piece out the different components of what parts of the neuronal activity are required. And sort of we had some clear predictions, and because, for example, we know that these synapses release glutamate, and that glutamate uptake into the astrocytes through the glutamate transporters drives a current that should be depolarizing, sort of. So we would clearly predict that glutamate and glutamate uptake should be a part of it. And so we essentially, in figure 6, go through a lot of pharmacology to really piece out what's required, and sort of we clearly need presynaptic activity. Without it, we're not directly activating the astrocytes with a stimulator. So that was sort of more of a control. And where it got really, we did the glutamate transporters with the blocker TBOA, which essentially blocks all of the glutamate transporters, and that clearly affected the peak of the depolarization, pretty much as we predicted. But sort of the notable thing was it wasn't the primary component of it. What looked to be the primary component is actually potassium release from the neurons. So we tested this by either overexpressing the astrocytic potassium channel KR4.1. So this is an inwardly rectifying potassium channel that's critical for potassium buffering by astrocyte, and that's been explored in the astrocyte literature for a couple decades now. And so we either overexpressed it to sort of enhance the potassium buffering, or blocked it with a low concentration of barium. And that bidirectionally modulated the depolarization, and by a decent magnitude. Now, this also brings up a sort of interesting component that had us scratching our head a little bit, of when we clamp an astrocyte and record the potassium currents through KR4.1 at the soma, we get a depolarizing current of potassium flows into the astrocyte, and we get depolarizing current. Whereas when we look at our voltage imaging, when we block the KR4.1, we actually get a bigger depolarization. So at the soma, when we record it, if we block it, we block the depolarization. But in the process, in these out of the processes, it enhances it. So it really goes the opposite direction. So what we think is going on is that there's actually the potassium is doing two different things. One is a current through these potassium channels into the astrocyte, and that's what we see at the soma. So when potassium flows in, it's a depolarizing current, and we see that at the soma. What we see in the processes is that extracellular potassium builds up, and that directly depolarizes the astrocyte. So essentially it changes the Nernst potential, or the potassium reversal potential, and that directly, rather than through a current, just sort of directly depolarizes. So that's sort of how we sort of understand this, that we see different effects of the potassium in the KR channels out in the processes, versus what we see traditionally at the soma. Now, one of these sort of elements to the potassium that was really surprising and sort of still has us scratching our head a bit is that the postsynaptic receptors, so the NMDA receptors and the AMPA receptors, don't seem to contribute at all to the depolarization. So we tested this by essentially doing a control or blocking, or in the same cells, blocking the receptors with well-established pharmacology of DNQX or APV, and really had no effect. And this was sort of really surprising, because we figured, and sort of the literature sort of figures, that these postsynaptic receptors contribute a lot to potassium buildup, because they pass potassium, and they probably should pass more potassium than what's coming off the presynaptic side through KV channels. And honestly, we don't have a complete understanding of what's going on with that, honestly. And there's some really interesting work that came out recently from Matthew Parsons, that pre and postsynaptic sides of the neuron really see differences in astrocyte function, and sort of probably to differences of astrocyte interactions, and that this really could be contributing that these astrocytes maybe are highly biased towards the presynaptic side of the synaptic terminal. And that's sort of the best understanding and explanation we have there. But it's something that we're absolutely interested in, would love to sort of figure out how to get at that, and get at that better and more. But so sort of to put this together now, of how we see the whole picture, is what we think is going on is neuronal activity on the presynaptic terminal is releasing potassium into the extracellular space. The thing which makes this work is that the extracellular space, especially around the synapse, is very, very small. So there's really nice YAMs of how astrocytes wrap around the synapses, and you can sort of really see that there is very little space there, so that even small releases of potassium will increase the concentration there quite a bit. And sort of we think the concentration is going up by close to 10 millimolar, which is sort of huge on the potassium literature. But the point is that this is a highly, highly focal buildup of potassium, that this potassium will, in a fairly short time, tens to hundreds of milliseconds, will diffuse away or get taken up by the astrocyte, and sort of you never really get a bulk increase of potassium, which sort of all the potassium literature is just mostly based on sort of potassium sensitive electrodes that are sort of put into the tissue slice, and that sort of records much more on these sort of bulk global changes. And they really don't see big global changes for the levels of activity we see. When there's seizures and ongoing sustained activity, they get up to 10 millimolar changes, but that's really under seconds to minutes long ongoing firing. So really, what really think is sort of like there's these highly localized pockets of potassium buildup, and they then depolarize the astrocyte, which we think then like feeds back onto the glutamate transporters, and there's probably a lot of other downstream targets of this. But we focus on the glutamate transporters because there we have the best handle on and the best tools to assay it. And sort of we see that our manipulations of our depolarization sort of mimic themselves with effect on the glutamate transporters of hyperpolarizing it enhances function, depolarizing it decreases function, and that is sort of like lines up and makes sense. So that's sort of like where we left this study, and I think this opens up many sort of new questions to me of like what's really going on in these like tiny pockets of like extracellular space around these synapses, both in terms of like potassium buildup, of like glutamate diffusion, clearance, and how this potassium is going to affect not just the astrocyte side, but the neuronal side of the neuronal side is probably going to be affected, and should there should be signs on the neuronal side that there's focal changes of potassium around it. It's again sort of a question of getting these GEVIs and these tools into the presynaptic side, and sort of like getting the assays working of trying to figure out like what there is the most sensitive to changes in extracellular potassium. But on the astrocyte side, there's nice studies and reports that suggested that, for example, that L-type voltage-gated calcium channels are expressed and functional in astrocytes. And sort of this was always one of the questions of when would these ever get activated, and sort of like this at least provides a putative mechanism of like that we get large depolarizations large enough that it could potentially activate voltage gated channels, and suggest that maybe there's much more going on in these astrocyte processes than we've been able to sort of like record or like appreciate so far.
Thank you so much for this, amazing presentation or work that you've been doing. It's so interesting, and it leads me to so many more questions, and that's why it's really cool and amazing. So, are you planning on maybe combining either very local uncaging of like glutamate on specific synapses, or using some optogenetic tools with very precise lasers that you can just elicit specific synapses like glutamate or activity in very specific synapses, and see what happens in the astrocytes, because there are synapses that prefer to end up on the boutons, and then there are synapses that prefer to be on shafts, and also more distal, more proximal. And we kind of know that these generates different upstates and different type of threshold, like changes different threshold levels in neurons. Could you imagine that there are also different types of presynaptic sites that generate different pockets of um of
Yeah,
change in the astrocytes? Yeah.
absolutely, and I completely agree with your thinking that we already know from like really nice EM studies that astrocyte like ensheathment of synapses is highly variable. And I mean, numbers vary. I think in the cortex, it sort of, I think the number is somewhere around 50% of synapses have like a closely adjacent astrocyte process. The other half don't. And so I think there's that. There's these synapses onto like boutons versus onto shafts, or that both like in like excitatory cells, or like onto interneurons that really don't have the like spines. And I think that's something we absolutely want to go after, because like you said, sort of they really should be different. Their like astrocytic coverage is probably different. They're like whatever the ultrastructure is, we know is different. So, and I think a lot of this is highly dependent on the ultrastructure. So the tools to get at it are tricky. So we've tried a lot of optogenetics, and sort of a lot gets, it's not impossible, but a lot gets really confounding, because sort of, yeah, we would love to sort of like highly focally like depolarize an astrocyte, but there was sort of a couple nice papers that really established that pretty much all the like optogenetic channels like actually increase extracellular potassium a lot. So it's sort of really like a mix of really like figuring out what tools can we really get sort of a clean manipulation. So we're absolutely working on that, and that's absolutely directions we want to go. And I think some of it we can do by sort of targeting, like trying to label these types of synapses, of like focusing, say, on synapses onto interneurons, that these would be more shaft synapses rather than synapses onto excitatory neurons, which are probably more biased towards spine synapses. I think that's sort of one way to go at it. I mean, I think there's certainly other ways, but that's we're absolutely interested. I think there is clearly also presynaptic diversity. I mean, at a minimum, we sort of know that there's a lot of heterogeneity presynaptically for like release probabilities, how they're facilitating or depressing, and that might be reflected in their like KV channel expression, and their which could like tie into this. And I think that's somewhat that hasn't been explored as much, which of like, which is also like highlights that tools to look at potassium and changes in potassium are still really limited.
Yeah, so to develop those tools, I know I met Peter Higeman at a different conference. He's really nice and open to collaborate to develop like specific tools for biologists. He was one of the fathers of optogenetic, but he's still really doing a lot of cool work to like generate specific new optogenetic tools for different approaches. You should maybe contact him.
Okay.
Yeah, he's really nice, and he's like really open to, like, he makes now some that are far out in the spectrum to like get different wavelengths
Yeah.
that they don't overlap and stuff. But he's really nice and open to collaborate with really cool biological new questions. So I think if you contact him,
Okay.
he will. Yeah, he's really nice. And another question I had was, I was working on a project. The idea was that these pockets of different ions, like I was more on the chloride inhibitory side, that the extra synaptic, or that they could represent some type of extracellular memory, a short term or even long term. Do you think that these potassium pockets, accumulate? Like, do you have an evidence that it kind of accumulates and holds like another form of memory in the astrocytes, so that they are involved in like short-term, long-term memory through these potassium
Yeah.
accumulation pockets?
So, I mean, I think the closest what we have is sort of we still think that sort of at least a decent chunk of our effect on glutamate transporters, on how they slow with neuronal activity, is probably through this potassium buildup. And so we know that sort of persists on the order of 30 milliseconds or so. Sort of, we know that if we sort of space a stimuli longer than that, sort of it quickly starts to recover. So I mean, that sort of at least provides a very short term, sort of like much short term like memory effect of it. I think there's, I mean, I think one of the things that would be really exciting to me, sort of on the like memory effect, is also like how this affects neighboring synapses. And I think it's a fascinating question, but it's also something that's really hard to make tractable, because it's sort of, you would imagine that sort of this potassium is going to diffuse out, and sort of if you have it active enough, it's going to sort of at least very locally like spill onto the next synapse. But it's sort of or like depolarize the like astrocyte process a couple microns onto for the next synapse. But sort of the question is like, where is that next synapse from? Because now it's really what is in spatially like the closest synapse question, which is sort of something that I don't think people normally think about that much, because since they're like all intermixed and sort of in no way has to be from the same neuron, or even a close by neuron. It could be a completely different path, because it's really just like, I think what's closest in the tortuous shape of the extracellular space.
So I'm not sure. I'm not currently in the field anymore. But is there somebody working on a specific potassium indicator, like not general voltage or
There,
positive ions?
yes. So there were two that were published, at least on bioRxiv, in the last 6 months. One from Ed Boyden's group, and one from Yi Shen and Robert Campbell's group. And sort of, I mean, we've talked with both of them, and sort of both of them sort of say sort of that it's a really hard problem. And sort of what they've gotten is sort of they've gotten ones that might work okay intracellularly, and with various caveats of still pH dependence. Sort of like I think they're on the like cusp of really being useful. The sort of unfortunate thing that's really still missing is nobody seems to be able to get them to work extracellularly, which would be, for us, would be fantastic. But sort of, I know both those groups and another one in Austria have sort of tried, and nobody has managed to make any progress on that so far. And from what I've heard, is sort of like, on the tool side, it's been a little bit of a dead end, unfortunately.
Oh, okay. Would there be a way to just, I
I
worked
would
on
love to have those
Clomeleon.
tools, essentially.
Yeah, I worked on Clomeleon for a while, and we tried to with George Augustine, and then we tried to like tag chromatin to extracellular and intracellular like receptors, but then on two sides, like
Yeah.
on one the extra. But it kind of was challenging. The construct was getting kind of really big to express it and keep the cells healthy. But maybe there would be a way for potassium to just tag a FRET type of genetic indicator
Yeah.
to the extracellular side of a receptor.
The problem, as I understand it, is that once these sensors go through the ER, they seem to completely lose their function, and through the speculation is through palmitylation or some other modifications, but of essentially essential amino acids in the potassium sensing domain of it. So, I mean, we've gotten them to traffic extracellularly, but unfortunately, they all like lose their function.
Oh, okay. Yeah, sorry.
Yeah, exactly.
Went way too.
It's something we've been following closely, but it's sort of at some point we said we're not experts in developing these tools, and sort of if I mean Ed Boyden's lab and Robert Campbell's lab both do fantastic work in developing these fluorescent sensors. If they tell us that this isn't happening quickly, I somewhat take their word
Yeah,
for it.
for sure. Yeah. Okay. Well, I'm sorry I took over the stage. So Serena and everyone else, please go ahead and ask questions. Thank you.
So, well, those were fascinating questions. I wanted to pick up on well, a question you even raised. What in terms of the spatiotemporal effects of this potassium burst, and what are the neighboring synapses pick up, and do pathways get crossed? But I'm still connecting the so the synapse and that initial potassium burst, that's going to depolarize locally, but that depolarization still extends over microns, and so then it's not just the immediate neighborhood. Does the depolarization of the adjacent processes then affect, um, I guess ultimately you're looking at glutamate clearance rates in other synapses, or how does it affect other synapses? Because I'm
Yeah.
ultimately looking for how does neural synchrony get driven, and is this a possible mechanism for that?
So, I mean, partially it's we haven't found a tractable mechanism to test it, and sort of if we sort of stimulate two different pathways, sets of axons onto a single astrocyte, sort of we get almost no overlaps in the depolarization or on the glutamate effects. But that is sort of doesn't directly answer it, because sort of we don't really know like what the like average distance between those synapses are. I mean, that's sort of the question would really be is sort of can you find the closest adjacent synapse when, and that sort of there just isn't a labeling technique to really get at that cleanly, at least for us. So I mean, it's that's what's sort of like has been sort of like stopping us from like really going gung ho after that, because we just haven't had a clearly tractable experiment for it. But that said, I think it's an absolutely fascinating question, and I think, I mean, one way to sort of try to get at that would be sort of a bit of like computational modeling to sort of see like how big of a spread do you really need to affect something, I mean, there are at least like computational models of like diffusion or like
Hmm.
space constants that could be harnessed.
I see. So you really can't set the experiment up to know that two synapses on different pathways are close to each other.
Yeah, exactly. I mean, it's sort of a best case would be sort of like hunting in the haystack to
Uh
sort of
huh.
like I mean, sparsely label sort of so that you can pick up individual like synapses, and it sort of it might be possible in a very low throughput manner, and sort of in the end we've there have been more tractable experiments to keep us busy.
But then doesn't the time dependence of the same synapse sort of indicate? So if you're getting a decreased glutamate clearance rate, or increased
Yeah.
rather, um, near in time to the event, and that depolarization propagates over distances, wouldn't neighboring synapse have a modulated glutamate clearance rate, whether they're firing or not?
In
Katerina, you
depends
got a hot mic.
depends on exactly how close, and sort of I mean, I guess the other thing that it really depends on is the like astrocyte ultrastructure of is it really like a branch that comes out touches one synapse and a different like branch coming out touching another one, or is it sort of more sort of a bit like an axon of like one branch passes a couple synapses, and sort of like ensheaths one after the other. I mean, I think that would sort of also like give dramatically
Hmm.
different effects.
But you did say that was, but so those hot spots though that you imaged, those were over microns. So whatever
Yeah.
the bushiness topography is, there's going to be many synapses in that region that are all in the quote blast zone, right?
Yes, with so sort of I think our the like micron size might be a bit overestimation just due to like our microscope, because it's sort of like it's the like true depolarization size is convoluted with our like point spread function, so which is
Hmm.
going to make it look bigger, because we know sort of the like for example the width, our width that we measure is half a micron or so. Well, we sort of know that the width of like an astrocyte, the small astrocyte processes, is down to like 100 nanometers, so sort of we can't I think our estimation is probably a little overestimation just due to like imaging effects. But that said, I think it probably should spill onto a neighboring synapse if you can catch a close enough one.
Okay, so then I understand what kicks off the calcium waves is there's potassium calcium exchange. So does this initial potassium blast, if it's repeated in pumped, so there's an active neuron pumping a lot of potassium activity, does that build up as in kind of an integrative function and get the calcium waves going?
That is a good question. I mean, we haven't seen it. Sort of, if we take our same preparation and do astrocyte calcium imaging, we don't really get a repeatable calcium signal from it. And I think sort of unfortunately, there's a like plethora of things that initiate calcium signals, from like ER calcium to pumps to potential calcium channels to, so I mean, I think it's sort of like back to our like pre-discussion. I think a lot of it probably depends on exactly what state
Mm-hmm.
the astrocyte is in. I would guess that this potassium buildup can certainly contribute, but it's sort of it's not a one-to-one correlation of we build up potassium, we get a calcium wave at least. And so there's something more complicated to that.
Yeah, that strikes me as just glaringly interesting and mysterious, is that you can't even get a reproducible calcium wave, yet
Exactly. I mean,
they're all over the place. But yeah, what we
I mean, sort of, there's, as far as I know, there's almost no astrocyte calcium papers that have used a stimulator. And I think there's a good reason for that. And I mean, we haven't gotten
I think
anywhere
so
with the
too, whatever
stimulator.
that answer is. Yeah, but
So
okay.
I mean, there's clearly something interesting going on.
Uh-huh. Okay. I want to give some other people a chance to speak, but I'll have more if later.
Hi, Doctor. May I ask a question, please? First of all, that was an incredible talk. I only learned about astrocytes just being in the Science Society here, so I'm learning a lot here with this talk and with Serena's inquiries and than before. But my question for you um, is there a rhyme or reason to the sheathing of synapses by the astrocytes? Like, is there any sort of predictability to what they attach to and why?
That is a good question. So there, the ensheathment is certainly plastic. I mean, there's some good papers that sort of show that you can like retract them, and sort of they're also like respond to like neuronal activity through like long-term potentiation that I think, if I'm trying to remember it correctly, also retracts it. So I mean, it's definitely an active thing, and it's definitely sort of responds to activity in some manner. But I don't think we have a really clear understanding of like what differentiates an ensheathed synapse versus an unsheathed synapse in terms of their activity or any other properties. It's also partially in that like you need EM images to really establish the sheath to some extent.
I see. So as it stands at the moment, then all we know is that astrocytes do attach to things, to the synapses and things, but they can attach to certain areas more than others for no reason that we know so far. Yeah,
Yeah, exactly.
yeah.
There's a couple interesting manipulations have been like found. One really interesting one was these gap junction proteins. So these sort of like form like cell to cell intracellular connections, and if you astrocytes express a lot of these, and these sort of connect astrocytes in a syncytium. And one of the interesting and curious effects of knocking out one of these gap junction proteins was that astrocytes, for example, really pushed their processes like into the synaptic cleft. So like almost like super sheathed and sort of like tried to like go even further into the synapse.
Oh
And
wow.
so that's definitely like one of the like really curious and interesting manipulations of it.
That's incredible. Because I remember Serena, I believe, mentioned something about, was Serena Einstein's brain, that there was astrocytes, a higher deposit of astrocytes or something in the part of the brain that involved
Well, that was
spatial
a
awareness or
that was a finding out of the autopsy. Yeah,
yeah,
but no,
yeah.
it's fascinating that I mean I hadn't heard of that. That's so you block their gap junctions and cut them off from the syncytium, and they just drive further into the synapse.
Yes, this was. Let me pull it up. This was a paper from I think Natalie Roche, and it was I believe knocking out connexin 43. And yeah, it essentially tried to the astrocyte sort of tries to push into the synaptic cleft.
Oh, wow, that's a bizarre type of regulation there.
Yes, exactly. Absolutely.
I'm wondering if in some point in the future we might find that in terms of like our own health, we'll have to take care of, not just like, you know, brain health and body health, but like astrocyte health or something. The more we discover about these things, they seem quite remarkable. Thank you very much for taking my questions, Doctor.
So,
Are there others? Flash your mic if you have other questions. Oh, Dr. Shah.
Yes, thank you so much for the, fascinating research that you shared with us. So actually, I was just wondering about the clinical use of that, and a couple of the thoughts came to me. First of all, I just thought about some of the disease, for example, such as Huntington disease, that we have a, problem with the potassium, repolarization somehow, and is type of the neurodegenerative disease. Somehow in Alzheimer disease and the recent research that they had through the Alzheimer disease, they just found the function of the astrocyte and dual functioning of the astrocyte. So I was just thinking about, because you talk about the excess of the glutamate, extra glutamate and potassium level, and somehow that's not clear. And so in the clinical level, if you cannot inhibit that, and because you mentioned about the optimization, so do you have any extra, information that you can share with us in, some of the disease that might be related with your research?
I mean, I can point. I think sort of the potassium mechanism sort of definitely has a lot of connections, and sort of the one we're going after is sort of epilepsy. But I think there's many other connections. Alzheimer's certainly being one. There's, in terms of like really fascinating studies on sort of the potential potassium effects. One that I can point to is there's a study from Highland whose lab, that sort of really ties these potassium changes as surrounding neurons to depression. And that was a really exciting paper that came out a few years ago from her lab, really looking at like this KR4.1 channel that buffers a lot of the potassium in a completely different brain region in the lateral habenula. And I mean, Alzheimer's definitely affects glutamate clearance. And I think it's been a bit less studied on the potassium front, but sort of the like A beta peptide can, I think, directly interact with the glutamate transporters to inhibit them, for example. That's been published as well. So I mean, I think there's a lot of interactions, and sort of how all of these sort of like ultrastructure, that a lot of this sort of like depends on changes in these neurodegenerative disorders is, I think, also a really interesting question.
Yes,
Because sort of,
because when we are thinking about the glutamate cycle versus the, other side of that is a GABA, but you exactly talk about the potassium. You had emphasized on potassium and potassium in relationship with astrocytes. And that's why I was just curious more specifically about, diseases related with the astrocyte and the dual nature of the astrocyte.
yeah, no, I completely agree. And I mean, I think there's more and more work being done on sort of the like astrocyte component of these diseases and how it contributes. And I mean, I think that's definitely something we're trying to go after as well. Uh, uh, and it, but yeah, I mean, sort of, I don't have too much other to really like share on that front, and sort of our work is still on the like early days there.
Thank you so much.
Oh, hi. Thanks for this, wonderful presentation. And presenting this very interesting research on exercise. I myself also learned a lot. I have a question for to understand the work better. That the pathway specific keywords in your abstract refers to that so far seems to be. Can I take it as more a it's a responsive role playing by astrocytes to the upstream to the by the neuron, and then there isn't a correlation so far found. And in your conclusion, that the seems that there's. I'm curious the uh, the role at this point, yeah. So the clearing of potassium and ions and others. The what is the could you a little bit more? What is the specific potential for modifying the synapse plasticity? I mean, already we know that it plays roles in, say, repairing, and does it take a role on, say, like a neuron type of role? That's just maybe it's not uh, ask the right question, but just from my own learning.
So the pathway specificity is more what was this one experiment where sort of we can put two stimulators in our brain slice to sort of activate ascending versus sort of lateral like inputs, and mostly to show that sort of the depolarizations or the glutamate effects that we saw were didn't spill from one stimulator's effects onto the other, sort of that. And we got the hotspots we found were non-overlapping, or the glutamate effects didn't spill from one pathway to the other. More or less sort of like trying to get at sort of the question of like, is it a larger area or that is sort of spilling on, or is it highly focal? And sort of to the question was like, can this sort of be modulated and sort of like modulatory? I I think so. And I mean, I think partially to some extent it's novel territory to go after of what like this potassium buildup actually does to the neuron, and sort of you'd think that sort of like it could affect presynaptic release for train different patterns of activity for sure, but it's something that hasn't really been explored yet, because I think just because the effect was unknown. And I think there's definitely potential of like interventions of sort of, if we get a better handle on how astrocytes clear the potassium, that could give a new crank to turn on changing this buildup one way or the other, and to sort of use that as a tool to modulate or change the neuronal states.
Thank you.
Well, wouldn't the impact on the glutamate release impact the postsynaptic? Is there a coupling between the postsynaptic potentiation and the rate of clearance of glutamate?
Surprisingly, the like postsynaptic side seems to be somehow uncoupled from it. So there's a really interesting paper out very recently from Matthew Parsons in Canada, essentially looking at glutamate and then glutamate clearance and like what the presynaptic side sees versus what the postsynaptic side sees, and sort of the presynaptic side seems to be much more sensitive to any disruptions, while the postsynaptic side seems completely invariant. It's sort of, there's clearly something interesting going on there. We just don't have a good handle on it.
Was thinking about this recently. There's some recent work suggesting that there's a little more compartmentalization going on in the cleft than maybe we realized. Like one of the lines of effort is about transsynaptic proteins.
Yeah,
And yeah, go ahead.
yes, I mean,
Well, yeah.
there's been really nice work sort of showing that there is like transsynaptic alignment of release sites to postsynaptic receptors. And it's one of the really, really nice things that has come out of sort of advances in super resolution microscopy to some extent.
So with this triune synapse, my thoughts go to where exactly are is this action happening in terms of, because I know there's a lot of work showing in strongly coupled interactions between astrocytes and the presynaptic terminal, and then the question is, where exactly are the receptors that are getting signals from astrocytic release, and like this kind of spatial detail might be critical to talking about why it would be more pre versus post synaptic.
I completely agree, and that's our thinking as well. It's just on a size scale that makes it not untractable, but not easy. In that, I mean, we're talking on the orders of tens of nanometers of differences that probably play a critical role.
Well, I did some work a few years back on the cannabinoid receptor, and it's the endocannabinoids, arachidonic acid, more in the brain, but in the peripheral it's more 2-acylglycerol. But it's would function as a retrograde messenger,
Yeah.
and in the astrocytes express a cannabinoid receptor as well. It's curious. I mean, you mentioned it. There may be some differentiation in the astrocytes as far as what gets expressed presynaptically versus postsynaptically. Or I wonder if it's a temporal effect that the postsynaptic is releasing the arachidonic acid, and that's actually activating those cannabinoid receptors both presynaptically and on the astrocytes. And if that has some modulation, it would be interesting to see.
Yeah,
It's
no,
been studied in
I
neurons.
completely agree.
Go ahead.
Yeah, no, I completely agree, and I mean, it's something we've been aware of, and sort of we dip our toes from time to time, but sort of we haven't had something jump out enough to sort of pursue it at the moment. But I think it's definitely an interesting thing, interesting aspect, and something to follow up on.
So it's fascinating that you've and in please, anybody, just flash your mic if you have other questions. I'll just keep going. So you're saying, or you found that locally that depolarization is up to 20 millivolts on the astrocyte, but when you patched the soma, you'd only it was more like 2. It was much less. Is that like the soma is really just sort of a buffering kind of activity, but all of the actual signal is local and very temporal?
Yes, to some extent, sort of. It's also soma depolarization is very different mechanistically. So the soma depolarization is purely a current. Is purely like a inward potassium current causing it. So if you block the inward potassium current, there's very little left at the soma. So it's really sort of a split between the soma sort of like integrates like the current coming into the astrocyte from the a lot of the astrocyte or all of the astrocyte, while the processes, the depolarization is mostly sort of the extracellular buildup of potassium. So that's really sort of the like split between the two of like the extracellular buildup of potassium is never spatially big enough to sort of ever get to or affect the soma, because the soma is sort of away from, not like touching a synapse.
So really the signal is, it's not the influx of potassium, it's just the voltage depolarization of the temporal event.
Exactly. It's the extracellular buildup rather than the influx that's causing the processes to depolarize.
So this leads me to a question: is there a more localized and gene expression modulation and mitochondria out there? Are they more focused on the?
There's mitochondria out there which sort of like barely fits into these processes. There's there was a paper 5 years ago now that did sort of localized gene expression in the processes versus the soma. I mean, I'm vaguely recalling it. I think there were some differences. I forget exactly what jumped out from that assay. I mean, I think one thing to sort of appreciate as well is sort of a lot of these proteins, especially the glutamate transporters, are very highly expressed. And they're extremely densely expressed on astrocytes. So there have been some really nice papers sort of like quantifying their density and their expression level, and sort of the two forms of the astrocytic glutamate transporters together comprise about 1.5% of the total brain protein by themselves. So you can already sort of picture that these are have to be packed in there incredibly densely.
Yeah. So my question kind of is, I'm thinking about if they, if the time scale of the depolarization in the soma is so short, and I assume there's not much calcium release, that like, is there some sort of, how does gene expression change to adapt to, environmental changes, and the environmental change are other neurons and stuff like that. So like, if
Yeah.
it can
I mean,
be
I
much
guess trying to
soma.
get at what the like downstream signals
Yeah,
from
exactly.
this is.
From this activity, like, because
Yeah.
the ATP production changes based on, a lot of neural activity around them, like a lot of potassium or not. Like, yeah, I don't know. Is there a good thing? Is there a way to monitor that, maybe? Like, local ATP production, is there an indicator for that? I'm sorry for keep asking.
There are some they're mixed of mixed usefulness. I mean, there I haven't followed that literature as closely. I mean, I think that's definitely would be interesting, and then I mean, a lot of this is but has to be like energetic of like the role, especially with the like sodium potassium pumps, which get highly co-localized with the glutamate transporters. So I mean, there has to be significant ATP demand there for sure, and that could definitely signal. Um, yeah, I mean, I don't think anybody's really tried to do that yet. But
Oh, that would be really interesting, I think, to like see local ATP,
yeah, I agree.
how long they exist, maybe, like with, let's say, I don't know, with long-term depression versus if there's something going on like that, and if it would reflect maybe in local ATP production, that would be cool.
Yeah, no, absolutely. I mean, I think the whole metabolomic cycle in the brain is sort of getting more and more attention as well of that this is highly regulated and highly dynamic.
So there's a brain initiative grant to make a good ATP indicator.
I mean, the ones I know is there's one that uses luciferase to sort of my PhD lab was using it to sort of do bioluminescence to measure ATP. And then there's a couple FRET ATP ADP sensors that are out there. So I mean, there are some at least starting points for these tools.
So now, when you said so during the depolarization, is that glutamate clearance rate increasing or decreasing?
Decreasing, it gets slower.
It's slower. So um, the more active that presynaptic neuron is, the less efficient glutamate will be cleared.
Exactly. It's a counterintuitively a positive feedback loop.
Right. So that serves to amplify the signal at the postsynaptic side.
Exactly.
Hmm. And which is really cool. And
Unless you get a lot of inhibitory neurons, you know.
right, right.
Yeah.
So
Yeah.
does this work the same way for the astrocytes that are expressing GABA receptors?
So GABA transporters are clear GABA. So GABA transport is generally a lot slower than glutamate transport. And it's sort of, as far as I understand it, it's halfway split between GABA getting cleared by neurons versus getting cleared by astrocytes. So it doesn't seem to be similar. It definitely has a different modulation than glutamate, so it doesn't have the same like positive feedback loop. That said, I think it's been a lot less studied than glutamate clearance, so there may be things that we just haven't found yet.
Well, don't the astrocytes also they also express the GABA B receptor, right?
Yes, they also express the GABA B receptor. I don't know honestly that too much about what comes downstream from there.
Yeah, and that may be regionally specific. I remember reading a paper about the medial prefrontal cortex interneurons. But you
I
were
think it's
studying.
also like in the hippocampus. I think the
Mm-hmm.
GABA B expression is fairly broad. It's sort of it's been this sort of like perpetual questions in astrocyte of they seem to express a lot of stuff, especially if you like look at the expression studies. So that there's like NMDA receptors, the GABA B receptors, and sort of really get nailing down a reproducible function to them has been
Yeah, they
tricky.
listen to everything.
Exactly.
Well, but so you were studying cortical neurons, right?
Yes,
Yeah, and because there's different morphologies, right, of the astrocytes depending on where they are.
absolutely. So I mean, the like glutamate effects of it, they're another lab, the Parsons lab, and like looked at different brain regions, and the hippocampus is much more resistant to it, for example, and but the like cortex and the striatum were both have this like I think slowing of glutamate clearance to a strong effect.
That's interesting, especially also in the striatum, because there's so much inhibition going on in the striatum. It's crazy. So it's interesting that the astrocytes are probably the only thing that kind of counteract
Huh, okay.
that. Yeah, it's like even the output is inhibitory, like like the projection neurons express. Like it's it's funny, but um, the interesting part is about the GABA B receptors because they're G protein coupled, so they
Yeah.
kind of are supposed to introduce like some intracellular signaling and maybe change gene expression. So yeah, it would be really cool to locally. So I was at the conference and there was like this German lab, which I forgot, I have to look it up, that was working on very local optogenetics that you could stimulate, but in a culture mostly with like nano LEDs very locally and change gene expression very locally with like nano
Oh, that's
LEDs.
cool.
Yeah, so you could basically at the synapse inhibit or activate specific genes, like let's say GABA B, and stop like expression over time just there. It was through the love LOV.
Okay. I think I've come
You
across them at some point.
you could probably do this with an optical strategy too,
Yeah, with
and you would.
so they had like strings of nano LEDs. They could program them without having cables and whatnot, because they had this tiny uh, receptors for signaling. So you didn't need a fiber into the culture. You had the strings of LEDs, and you could program them to, let's say, have different patterns of lights on and off throughout the culture, basically, like along the dendrite. It would be cool to also do that in astrocyte.
Yeah, that would definitely be interesting. That's a cool assay.
Yeah, I have to look up which lab it was and then what they did with it, because they were kind of looking at the conference for labs that would kind of apply it more. There are all these bioengineering labs, a lot of them in Germany, and they kind of are looking for collaborations to apply all these new fancy tools. So that
Yeah.
would be cool. So, but it's in a culture, and you do acute brain slices usually, right?
Yeah, so astrocytes, I mean, essentially the issue is that a lot of this really depends on that ensheathment, like ultrastructure around synapses, and that just isn't replicated in cultures for the most part. Sort of astrocytes get a much flatter, less ramified morphology in cultures.
Is it extra complicated to work with them in slice, like above and beyond what we do to keep neurons alive in the slice?
No, not particularly. I mean, they
Okay,
don't last quite as long as neurons for like good patching, and then patching astrocytes is a little bit finicky, but all in all, they're easy to
so
handle.
not much worse than dealing with interneurons, maybe something like that.
Yeah, no, not particularly worse. If you can patch interneurons, you'll be able to patch astrocytes and image them. Imaging is might even be easier. Sort of the the neat thing is that they tile, so they don't overlap, which
Oh,
is
that's awesome. That's
advantageous.
a dream. Yeah.
Exactly.
Uh, and I'm assuming, I just haven't thought about this in detail. I'm assuming there's quite a few, maybe like GCAMP6 lines for astrocytes, and I'm wondering if how specific in terms of different cell types, between
So
among the astrocytes.
yeah, so there's there's a couple GCamp mouse lines. They've honestly fallen a bit out of favor in preference of viral work, just out of GCamps and everything else is advancing so quickly. The mice can never
So
keep
yeah,
up.
same for neurons. We're doing viral GCAMP8 just because we don't want to deal with 6 anymore. But
Exactly. So, I mean, there's virally, unfortunately, there's a little bit limited promoters to work with for astrocytes. It's pretty much only this modified GFAP. That said, it expresses fairly specifically to astrocytes whenever you express like a sensor. There's so I mean it's fairly straightforward to express the newest GCamp. They usually have them straight at add gene, like a GFAP version of a lot of the viruses.
I have a question on the compartment, uh, compartmentalization part mentioned earlier by Wisdom. That the so I understand that your work is focusing on the cortical distal type of astrocytes. So within the, or even more general glia cells, the at other locations, say hypothalamus, that would it be possible to think that at the lower level that they take on a more active role, say more modulation that they can participate? Would that be a meaningful question to ask?
I mean, I don't know too much about hypothalamic astrocytes. I mean, I know they, for example, they can retract or extend their processes, I think, much more than what's been shown in cortical astrocytes. But I mean, I would hazard a guess that yes, that they're probably going to be active players as well, uh, and would be interesting to go after. I mean, everything I know of them sort of like suggests that they already have as much or more active interaction and changes in morphology than cortical astrocytes.
No. Could you repeat the last statement? They don't, or they do?
I think they do.
I see. Yeah.
So I think sort of there's various assays which cause them to like shrink and retract from synapses much more than what's been seen in the cortex.
That's interesting. Thank you.
I'm going to have to say goodbye really soon.
Yeah, of course. I was about to ask. You know, you've been going for a long time, so yeah. Thank you so much for
could I
this.
ask one last question?
Oh yeah, go ahead.
With these retraction events, has it been seen for them to return to synapses, or is there like a rate in which the triune synapses are replaced, or once they retract, is that usually the end of it?
I think they come back, but I'm honestly not completely on top of that literature.
Yeah, me either. That's why I asked. But it's really interesting to me because I study dendrites, and these retraction events are like, uh, without getting into the causes or functional role of it or anything like that, the effect certainly is to reduce specificity. So you get this spillover, and you could have these plasticity induction events that are now affecting multiple synapses on a branch, and synapses that weren't involved in the induction process explicitly. Yeah. So and if they come back, now we have this really interesting dynamic system for potentially clustering synapses on branches and things. I've been thinking about it in that light.
Yeah, I completely agree.
Does ARC influence that at all? Do you know? Some more?
Does what? Sorry.
Does ARC influence the retraction at all?
I was gonna ask about what triggers the retraction. Actually,
I
I didn't.
know
Sorry,
on a
go.
mechanistic level.
So not a terrible question then.
Yeah.
Yeah, that's really interesting. So does the retraction, like a faulty mechanism in retraction, contribute to epilepsy then? Because you also are involved in epilepsy research, right?
Yes. Good question. Honestly, we don't know that much. I mean, on first, so at least in a couple models, no, but honestly, I'm not sure. It would be the answer.
Okay, cool. Interesting. Next. There's still a lot to know, so
Exactly.
nobody will be out of job here in your future. That's a good
Job
thing.
security.
Yeah. Don't solve too many questions for someone more.
Oh, there's plenty left.
Yeah, exactly. There's a lot left. So yeah, thank you so much for taking so much time and answering all of our questions and for sharing your really interesting research. I wish you all the funding and that you get a lot of helping hands.
Thank you, and thank you for having me.
Yeah, this was
Thank
so.
you. Yeah, this was fascinating. We could certainly keep asking questions, but we'll respect your time.
Yeah, maybe you
Thank you
come
very
back
much.
with some updates on your research. If
Sure.
you maybe,
Yes, do come back.
yeah, maybe there
Well,
will be a great breakthrough for potassium indicators.
yes, exactly. If that comes out, I'll certainly be back.
Okay, thank
Yeah,
you so
have a
much. We
good evening.
really appreciate it, and enjoy the rest of your evening. Bye, Moritz.
Bye.
And yeah, thank you everyone for being here and asking so many questions, interesting questions. And we really appreciate it. If you like discussions like this, follow the club, Science Society, and we will have, of course, more rooms in the future. We'll have actually tomorrow morning at 9 a.m. EST, Dr. Kiehi. He developed self-assembled logic circuits from proteins, which will be really interesting. Then we'll have Dr. Steve Kihu. He is talking about the expansion of desert climate in Central Asia. So it will be a climate change related room. And on Friday, we'll have Dr. Kagan talking about gene mutations across species and how they shed light on aging in humans. And yeah, this is our rest of the week. And yeah, thank you so much, everyone. Special thanks to Moritz again. This was an amazing discussion. And yeah, hear you all back soon, hopefully. Bye, everyone.
Thanks, everyone. Bye.
Thanks, everyone. Bye bye.